Do subscribe to Ekeeda channel and press bell icon to get updates about latest engineering, HSC and IIT JEE main and advanced videos.
Hello students, today we are studying chapter Human Health and Diseases. In that we are studying topic Acquired Passive Immunity. Now when I say acquired passive immunity, acquired means you have received, and when I say passive it means a second source. So what is this acquired passive immunity? Let's try and find out. Now when I say acquired passive immunity, it means an immunity that is developed from ready-made antibodies or already made antibodies when they are injected inside your body. What is this antibodies and how can they be ready-made or they can already be formed? Simple examination could be your vaccines. Those are the ready-made antibodies which everyone must be aware and which is very relatable. Let's come to a simple conclusion or a simple example for this. For example, every one of us has taken polio vaccines, that is an oral vaccines. All of us has taken BCG vaccines when we were a child, and those BCG vaccines have given us a small spot on our hand, especially in our upper part of the hand. Now BCG vaccines were given when we were small, and those were the first vaccines, and those first vaccines were nothing but dead organisms. Yes students, dead organisms. For example, if you are taking a polio vaccine, now polio vaccines are nothing but dead polio viruses, and those dead polio viruses, when they are injected inside your body, what happens is that your immune cells or your cells inside your body don't know that they are organisms. What happens is all your fighter cells are awakened. They think that it is a foreign organism invading your body, so everything gets activated in your body and antibodies are formed. Now these antibodies are against the antigen or pathogenicity of the organism. So if I say that antigen is the harmful substance of an organism, then antibody is the weapon that your body forms to kill that organism. And now this antigen antibody complex further carries out the process and kills and form other cells in the body, which is memory B cells, antigen presenting cells, and all of them. Your immunity in this form could be naturally acquired or artificial. Now when I say naturally acquired, how does antibodies are passed naturally? Remember when you were born, your mother, when you were a fetus, you were inside the womb of your mother. Through placenta, your mother passed those antibodies. And the second time is especially during vaccination. So we all have naturally acquired also and artificially also. Acquired passive immunity. Now when I talk about acquired passive immunity is specially developed when you develop or you receive ready to make antibodies. You don't have to make those antibodies, they are already given to you. It could be either form through an injection, which is an artificial form of giving it, or it could be naturally by your mother. Naturally acquired passive immunity. Now, when I talk about naturally acquired passive immunity, what I'm trying to say is these are antibodies or ABs that are transferred from the fetus or the baby who's developing in the womb of a mother through placenta. Remember a small cord that is connected to the baby as well as the mother that helps to pass on the nutrients, oxygen supply. Okay, through those cords, certain antibodies are passed, and one of those antibodies which we can name is IgG. That is one of the antibodies that your mother gives to the fetus before birth, and some antibodies are even passed through the colostrum after birth. Now, when I say colostrum is nothing but the thick, fluidy, milky secretion that a mother feeds to the child, the first milk secretion that a mother feeds. It is always advisable that a child should have that milk secretion because that milk secretion has antibodies that are given from the mother to the child. One of the antibodies could be IgE. When I talk about artificially acquired passive immunity, it means this immunity is developed after you're injecting previously prepared AB or antibodies using serum from humans, or sometimes it is animal. Now, what does this correlates to? Example, for a matter of fact, you want to prepare an antibody for a certain infection. It could be X infection. Now you want to prepare antibody for that. You take the antigen of that particular infection, and when I say antigen, I simply mean an killed organism, and you inject it in another animal which has similarity to be human beings. Once that is done, it could be lab mice or guinea pig or anything of that matter. Now once that's done, what happens is that particular organism or that particular animal will produce antibodies, and those particular antibodies can be clearly seen after a certain point of time in their serum. Serum is nothing but whenever you remove the clotting factors minus the RBCs from your blood, what else removes or what else remains is the whitish straw color liquid, that's serum. That serum material will definitely have antibodies to it. And once you found out those antibodies, what you can do is you can take out those antibodies from that serum material and further give it to different patients for different application process. So students, this was about antibodies and how antibodies can be injected to different patients. These were the two different types which we came across. Students, I hope we are now very clear that what this kind of immunity can do and how does that function. Thank you.